In this video, we will be doing negative staining with Nigrosin. For this experiment, you will need gloves, paper towels, a China marker, 2 slides, a test tube, lab coat, goggles, Nigrosin stain, inoculating loop, plastic pipette dropper, Bacterial incinerator, slant culture, germicide, slide box, and a biohazardous sharps container. In this video, we will be doing a negative staining with Nigrosin. Here's your slide. Here's your Nigrosin stain. And here's your plastic pipette dropper. To start, take your plastic pipette dropper and get a few drops of Nigrosin. The Nigrosin stain has a negative charge, like the bacterial cells, which also have a negative charge. When the stain is applied, the cells repel the stain. Now, you want to put a small drop of Nigrosin near the right edge of the slide. Now flame the inoculating loop. This will sterilize it. You can hold the loop in there for 10 to 20 seconds. Never just leave the loop in the Bacterial incinerator. It could destroy the loop by melting it. Next, get a small amount of culture onto your inoculating loop. This is your inoculating loop right here. Always pick the tube up by the glass, not the cap. That way the tube doesn't fall out and spill your bacteria all over the place. Pick the tube up with your non-dominant hand and remove the cap with your pinky finger of your loop hand. Never set the cap down. Keep holding the cap in your pinky finger. That way other bacteria doesn't get onto the cap and contaminate the culture. Flame the opening for about 3 to 5 seconds. This prevents unwanted microbes from getting in. Next, remove bacteria from the culture tube using the inoculation loop. Flame the test tube again for 3 to 5 seconds. Put the cap back on. Then set the test tube back down. With your inoculating loop, mix the culture gently into the drop of Nigrosin. Mix it from side to side. Then, immediately re-incinerate your loop to destroy the excess bacteria. Now you're ready to spread out your dye. Take your second slide, put it on your base slide at a 45-degree angle, and then pull it towards your Nigrosin dye drop. Then push the second slide away from the dye drop. This will spread out the Nigrosin dye. Place the spreader slide into a biohazardous sharps container. The spreader slide is contaminated with bacteria. Then let the slide air dry completely. As water evaporates, the dye bonds the bacterial cells to the slide. Once you've let your slide air dry, you may now view it under the microscope. Make sure when you're viewing it under the microscope that it is under 1000x magnification. The negative stain should look something like this under the microscope. Now, decontaminate yourself and your workspace. First, dispose of your gloves. You want to put these in the biohazardous waste container. Then use germicide to wash up your workspace. Once you've done that, use the paper towels to wipe down your workspace.